डॉक्टर साहब कहीं से आप करते हैं स्पीकर दबाना पड़ता है दबाना पड़ता है कि नहीं? तो डॉक्टर साहब यहाँ कैमरे में आएंगे आप इधर आके आप अनाउंस बिस्मिल्लाहिर्रहमानिर्रहीम अस्सलाम वालेकुम। First of all, I would like to thank Professor Dr. Ghulam Musharraf Saab for organizing this event and giving us all this opportunity to exchange knowledge and improve and enhance the chances of making our science and efforts more result oriented. So thank you very much, Dr. Saab, and thank you to every contributor and participant to this event. Today in this session, the first speaker is Dr. Madiha Isra. She seems to be very brilliant scientist with expertise in the molecular biology and genetics. After her graduation from Middle East Technical University, she did from a very high-ranking University of Houston, her PhD in 2012. Then she worked in Tuba Tech Research Center for Food in Food Institute as a chief scientist since February 2013. She is heading the Food Molecular Biology Laboratory and focusing on the DNA protein-based detection and analytical system and analysis. So therefore, I'm going to invite Dr. Madiha Isra to please share with us her expertise and experiences so all we can get benefit from her experience. Thank you very much.
Assalamu alaikum. You can hear me, right?
Yes.
Okay. Assalamu alaikum wa rahmatullahi wa barakatuh. Dear distinguished participants and all sisters and brothers, welcome to the first talk of today, and thank you for being here. It's very early in Turkish time though, it's 7 a.m. and I am participating from my home, and my 3-year-old son is still sleeping, and we should just hope that he will sleep throughout the presentation. So today I will talk about our method on detection of porcine DNA from processed foods and food additives. So let's start today with a verse from Quran. So there are many verses in Quran about pork, but let's read the one in Surah An-Nahl. Allah says that He has only forbidden to you that animals, blood, the flesh of swine, and that which has been dedicated to other than Allah. So Allah forbids us from swine, and we obey Him. So yesterday, actually, there were many talks about detection protocols, detection methods for pork detection, lard detection, and so on. But today I would like to focus on the processed food, processed byproducts of pork, which we cannot use the same methods for the detection. So what are these byproducts? They can be fatty acids, glycerin, gelatin, collagen, L-cysteine, kind of like the other also amino acids, enzymes, minerals, and protein sources. And if you realize, actually, all these byproducts are very purified materials. They are composed of very purified materials. So that's why after this high processing, they are purified and they lose many of the proteins or DNAs and other molecules regularly found in meat. So besides use them in they can also be used in cosmetics and medical area too. So what happens in this processed foods? So they are actually produced under high pressure, acid and or alkaline treatments, and sometimes heat and cold treatments and fermentation. And there can be some others. Those processes causes DNA fragmentation. So from meat samples, we generally obtain very intact DNA, which we can dissect kind of like much more easily. But from those types of byproducts, the same methodologies cannot work. So if you check the current situation, we know that the methods are generally either DNA-based or protein-based. And DNA-based methods are generally on PCR, and protein-based methods are generally ELISA-based. But what are the problems with these methods? With these methods to be used in high processed food? So as I said, DNA is mostly fragmented and broken in high processed food. So we cannot use the same PCR methods that we use for meat samples. And proteins are generally denatured after this high processing of heat, pH, or high pressure treatments. So the same ELISA methods cannot work for a high processed food. And another point is proteins like gelatin do not have distinctive characters between species. So it's like porcine gelatin is almost having the same sequence, amino acid sequence with bovine. So it's hard to obtain kinds of like antibodies against those gelatins and other natural proteins. So it's really hard to get a method with a protein-based methods. So what did we do? We want to have a forensic approach, especially for the DNA isolation. So we looked for a forensic DNA isolation method, and we checked we evaluated the real-time PCR analysis by selecting short target DNAs, which will be very highly repetitive. And of course, it has to be species specific to pork. And for the real-time PCR, instead of using cybergreen or other bulk detection methods, we have to be specific with Tuckman-based probes. So it will be more specific. So as if we are gonna develop a sensitive method, we have to use the Tuckman probe. So at the end, actually we used 4 or 5 different DNA isolation methods, and also we tried their combinations too. And at the end, what we ended up with for the DNA isolation part, there are actually, we can say there are 4 pillars of DNA isolation from high processed foods. The first one is you have to use liquid nitrogen for the greening, for the homogenization part. Because those samples, since, as I said before, they are generally composed of very purified materials, they have to be physically broken down, and the leftover, the broken DNAs that are hide inside those molecules have to be free outside in the buffer. And as the lysis buffer needs to contain high concentrations of SDS detergent and DTT reducing agent. So this has to be really high concentrations. Thirdly, phenylchloroform isoamyl alcohol extraction is actually required. So we want to skip this step, but when we skip it, it's not that effective. And finally, precipitation of DNA must be done by ethanol. Ethanol precipitation is a must. Commercial kits are generally using spin columns, and spin columns actually keep some of the DNA inside them and just try to have more purified DNA. That's their purpose. But since in those high processed foods, we just have very low concentrations of DNA, if we use spin column, and it's all fragmented, it's gonna all go around the spin column, and you're not gonna be able to elute any DNA. So ethyl precipitation is another pillar of DNA isolation. So when we move to the real-time PCR, we chose a DNA region called MPRE42. It's a porcine representative element. And it has a high copy number. It's actually a non-coding region of porcine genome. And it has no homology between bovine. And we selected a 95 base pair long PCR product amplification. So it is a short target DNA, and we designed a Tachman-based real-time PCR. So by choosing these methods, what we were able to see is we were getting very nice, this is the PCR results, by the way. We were getting very nice results from 100% pure porcine gelatin. And for 1% and 0.1% porcine gelatin, we were also getting very nice peaks. The signal was nice. And with bovine gelatin from different companies, and for no DNA samples, the negative control, that means we just use water. The PCR didn't give any false negative signals. So the method was working. And actually, we just recently published this method. It was in Food Additives and Contaminants Part A. It was actually just published in March. So I would like to talk about what we have more in this paper. So actually the crucial part was we compared our method with some of the commercial methods. We called our method as the Tubigal method, and we used Biotecon and RBO Farm commercial kits as the reference ones, because they were also saying that they are very sensitive porcine DNA detection kits. So we had some dilutions of porcine gelatin from 5% to 0.01%. And our method was able to detect all of these reference porcine gelatins, all of these diluted porcine gelatins. And Biotecon method was able to detect up to 0.1% porcine gelatin, and RBO Farm was not able to detect at all. And we had some packaged high processed foods. We bought them from Europe, and we know actually that they were containing some porcine byproducts inside. And with our method, we were able to get a signal almost from all of these samples. But the Biotecon kit was only able to detect one of the gummies. So our biofarm was not working at all. So the crucial part actually here, that we changed the DNA isolation method for all cases to our own method. So DNA isolation of Tubigal method was used, and then RT-PCR of other commercial kits were continued to be used. So this is, the first column is the same, our method. When you look at the Biotecon method, you can see that with the usage of our DNA isolation method, they were able to see up to 0.01% porcine gelatin. So the signal was coming, the signal increases like 10 times. So this is a very, very good explanation that DNA isolation is the crucial part. Of course, you have to have a very sensitive real-time PCR, but if you don't have the correct DNA isolation method, and if you cannot get the DNA outside your sample, you cannot detect anything. And you can see that from the samples, Biotecon method was this time able to detect like 5 more samples, only 3 were left over. So the detection sensitivity of Biotecon was increasing. And we also, like with this case, our biofarm was able to detect one of the gummies, but it was not working at all. So we did some validation studies with these methods to show its stability. For the parameters of DNA isolation, we selected the concentration, purity, and absence of inhibitors. So we selected very different types of food, from like jelly beans to juices to gum, like crystal sugar, chocolates, and so on, like very broad range of high processed foods. And we measured the DNA concentration, and you can see the absorbances at 260 nanometers. In general, DNA concentrations calculated are kind of like okay, but the main part is normally you have to make sure that DNA is pure enough and for this 260 to 280 ratio and 260 to 230 ratio has to be at certain ranges. This first ratio has to be between 1.8 to 2, and the second ratio is required to be between, it has to be greater than 2. But you can see that like none of these ratios are between 1.8 to 2, and none of these are greater than 2. So that means our DNA is not pure enough. There can be two reasons. Either these ratios are not, these absorbance values are not good enough to have a ratio. And the second one is our DNA is not really pure, because we actually know that we get them from very purified materials of food, and the DNA is actually broken and very in low concentration. So when we try to get it out, it comes with some other molecules like proteins, carbohydrates, and so on. So which is acceptable, like as long as the DNA is working in PCR actively. So the idea behind the DNA isolation is making the PCR work. So for that reason, we did an inhibition control reaction. So basically, in this reaction, you see whether the DNA you isolated can work in a PCR reaction actively. So we all had the positive results. So that means our DNA may be not pure enough, but it is actively working in PCR reaction. And we also checked for either animal reference or plant reference gene controls. For example, if we had the jelly bean, since it has the gelatin inside, we checked for animal reference control. For example, if we had like crystal sugar, it is plant-based, so we checked for plant reference gene control. So it was also actively working. But we have to see the origin, whether it's pork or not, right? So for that, we also had the real-time PCR validation studies. In those validations, we look for specificity, limit of detection, and inter-laboratory comparison parameters. And for the specificity test, we had some false negative studies and false positive studies. For false negatives, we selected reference porcine DNA, pork salami, porcine gelatin, jelly beans from porcine origin, chewing gum, apple juice, and aroma are added with 0.1% porcine DNA. And for false positive studies, we want to check the cross reactions, of course, beef, bovine gelatin, chicken, fish, lamb, jelly beans from bovine origin, and chewing gum, apple juice, and aroma. So out of 60 studies, we haven't found any false negatives. And out of 62 studies, we haven't found any false positives too. So specificity was working very nicely. So when we moved to the limit of detection studies, we had two different approaches. One approach was with the DNA copy number. So in this case, we had the reference porcine DNA, and we diluted it until 1 copy porcine DNA. And we had 12 samples for each case, and all in 1 copy, 5 copy, 10 copy, and 20 copy, all 12 cases were all positive. So that means our method was able to detect down to 1 copy reference DNA is actually DNA. So we don't, okay, this our method is sensitive, so this is showing that, but this is not actually indicating the samples, the high processed samples that we want our method to work on. So we move to the second approach of limit of detection. In this case, we used diluted porcine gelatins from 0.1% until 0.001%. We generally dilute them with bovine gelatin, and then at some point it just has to be diluted with water or some buffer. So out of 11 samples, we were able to detect until 0.01% porcine gelatin, but 0.001% diluted porcine gelatin was not enough for the validation study. So that means our limit of detection for this case was 0.01% porcine gelatin, or we can say processed food or food additive. So it was actually a very low dilution number. So that means the method was very, very sensitive. So we can detect actually contamination. So finally, the intra-laboratory comparison. We sent our sample, the method, with a blind to 3 different laboratories. We had 5 samples, gelatins, and also the DNA itself. All results were compatible from 3 accredited laboratories for more than 4 years, and it has been edited by our
There is some problem.
Can you hear me now?
Yes.
Okay, I'm sorry. I think there's some problem with my connection. So I just have a few more slides. So we have been accredited since 2018, and there has been more than 1,200 samples analyzed for porcine DNA. And actually we detected 30 of them as positives. So in a Muslim country, you know, so it's so sad though. So those positive, so these are actually from market studies. So we didn't select them from Europe, you know, so it's from the market, and we found them as positive in 4 years, 30 samples, and it was like not even completing most of the market. So those samples were including candies, chewing gums, biscuits, caramel-filled chocolates, juices like apple juice or sour cherry juices, like unfortunately, and of course some marshmallows and jelly beans. Oops. So there had been 2 national trainings given to outside laboratories and researchers in Turkey. And we had an international training by CIMIC. It was in October 2019. So we are trying to get our method available to everyone. That's why we had the paper published and we had the trainings. So I hope we can reach to many of the laboratories and let them do our method, let them perform our method. So thank you for listening.
Bismillahirrahmanirrahim. We are very fortunate to have Dr. Isra, because for living we need food, and for economic development and provision of within the Muslim country, this halal food certified, and to the Muslim not living in the West and not in the Muslim country, there these modern PCR based analysis are basically, I think, very, very precious and important development, and there is a huge scope in all the Muslim countries and in the non-Muslim country. So this is very amazing, and thank you very much. Now our next speaker is Professor Doctor. Oh, okay. Question. So therefore, now if the participant would like to ask any question to, then they are welcome. Oh,
Okay.
Mike.
Okay. So thank you for your detailed presentation. I am Dr. Wasim and working assistant professor at National University of Medical Sciences. So basically, I'm also targeting some biopreservative on these alternatives which are using as emulsifier and gelling agent. So my question is, what's your take on the bio-based product that are produced through the bacterial fermentation? Because these could be a good alternatives to the gelatin and other gelling and emulsifying agent used in the food and other FMGC products. The second question is, because the world is moving toward the bio-based solution and toward the more sustainable product, so we can produce these alternative in a better and cheaper way. Now these can be produced in a Petri plate due to the advancement of the technology. So what SMIC is an organization doing to do is joint ventures and also to develop the different R&D's products to focus these products. Thank you so much.
Thank you, Doctor. Am I wrong? I will just repeat the question. So if we can have an alternative of gelatin to be used in food, right? If that's so, actually there have been some work on it, but they cannot get the same gelling stability, even with fish gelatin. I think like consumers doesn't like the taste or the texture, so they still keep. And also like with the porcine gelatin, people say that it looks better on jelly beans. It looks much more how to say, it's more shiny, you know, like the porcine gelatin looks more shiny because it has more fat inside. So it depends on the consumers, though. If the consumers get used to different types of gelling agents, yes, they can be used in the food sector. Actually, our project with Dr. Musharraf, we just applied it and waiting for the answer. It was actually production of bovine gelatin in like from yeast with recombinant DNA technology, so that we can have it in another version, because it's hard to find enough bovine gelatin for all over the world. So it was another approach, but we are not sure that, okay, we can produce some bovine collagen, maybe like recombinantly, but we are not very sure that it's going to have the same texture. So if we can have the project, we're going to see that. So it's about they don't have very like distinctive taste, but the texture is the important one. So I think like there's one company in Germany. They were having this jelly beans, like vegetarian jelly beans, which were really people are buying them other jelly beans. But I think like it's not the same as the other ones, but they like the taste. So I think it depends on the taste, the consumer's reaction. I'm sorry that I didn't get the second question. Can you repeat that one?
So my second question was like SMEC is an organization. What they're doing like it is a joint venture to develop like R&D collaboration for the different projects to replace these controversial chemicals and other stuff, because EPS and xanthan gum could also be produced from the bacteria using the metabolic engineering and synthetic biology and other techniques like that.
Mm-hmm. You mean like SMIC can support us, you know? We have to ask Dr. Mohammed Ali. He's actually trying to get us some budget, but it is only for like inter-laboratory tests between countries. But maybe like pandemic might help in this case, because since people cannot move within the countries, they don't spend much money, so they can actually spend the money on scientific project budgets, they say. So I think like we should ask for such project budgets to CIMIC, and we should talk to Dr. Mohammed Ali.
Thank you for sharing your experience, valuable experience, and it is one of the best halal testing, regarding halal testing presentations. I want to know, you share one slide where you mentioned that validation through Qigam plus 1% for seen DNA. I need to know how it is possible. Please elaborate in a short words.
You mean how it's possible to detect the DNA?
Validation of DNA, you mentioned Qigam plus 1% for seen DNA. How it will be validated?
I am sorry. So you mean the protocol, or how we were able to detect that low concentration of DNA?
Protocol.
The protocol. For the validation, you have to have a specificity. You have to show the cross reactions and the types of food that you want to detect. So each case, you have to have at least like in total more than 40 samples have to be worked, have to be studied. And for you have to have a limit of detection study. And in that limit of detection study, you at least have 3 different dilutions, and like where you want to go as the limit. And if your limit is between the lowest dilution and the kind of like concentrated dilution, is much better. But for each case, you have to study at least 10 times. So to make sure, we did it like 11 or 12 times, but it has to be at least 10 times. And there are actually some other works too, but it was, let me just, I think I forgot one of them. Just let me check. And okay, we did inter-laboratory comparison test for PCR, but it was not necessary. It was just a new test that no one had before. So the accreditation committee wanted us to test it. Since it's a PCR, it's a kind of a known method, but it was not a standard method. It was not a published method when we accredited it. So they wanted us to show that at least 3 different laboratories can perform the tests. So these are the basic requirements. So you can add more on them, look for some other parameters, but these 3 has to be there for the PCR. For the DNA, you have to show the concentration, and of course the purity is always the problem. We are also having purity problems with the accreditation auditors too. Sometimes they don't accept it, but when we show that even though the purity is not enough, we can show that it's actively working in PCRs. And we also did like animal and plant references, so they cannot say anything because we show the actual idea behind DNA isolation. So these are the basic parameters.
Any other question, please? Okay, there is one request that please put your attendance in the given sheet. Thank you very much. Now I am going to invite Professor Dr. Abdurrahman from Indonesia. He is a professor of pharmaceutical chemistry at University of Gadjah Mada. He graduated from University Putra, Malaysia, in the field of with expertise in the halal food analysis. He is a very accomplished scientist with the uh, 250 publication, many book chapters, and uh, has been recognized in the form of many national awards. He is head of Institute of Halal Industry and System. So therefore, we are very fortunate, and I am inviting Professor Dr. Abdurrahman to please share his expertise with all of us. Thank you very much.
Thank Dokter. Assalamualaikum warahmatullahi taala wabarakatuh. Alhamdulillah, salatu wassalamu ala Rasulillah Muhammad ibn Abdillah wa ala alihi wa wala amma ba'd. First of all, I would like to thank the organizing committee for inviting me to share our activity on halal authentication analysis. And today I would like to present the analysis of percent gelatin in food and pharmaceutical product for halal authentication. As we know that the gelatin is highly purified protein obtained from the hydrolysis of collagen with high molecular weight, and also gelatin is one of the components commonly used in food, cosmetic, and pharmaceutical product due to its chilling properties. For example, this is the capsule cells made from the percent gelatin, and also this is the jelly coming from percent gelatin. As we know that from pig, some product can be derived from the pig, and also in the, for example, percent gelatin extracted from the bone and skin of a pig can be used as binding and gelling agent, and to ensure that the right texture is found in the gum and so on. As we know that the most commonly used gelatin in food and pharmaceutical product are percent gelatin and also a bovine gelatin. As we know that the presence of a percent gelatin is a serious matter for Muslim community, because Muslims are prohibited to consume any food or pharmaceutical product containing percent gelatin, according to the certain scholar of thought, yeah, or madhab. So the halal authentication analysis is intended to confirm that the product, food, cosmetic, or pharmaceutical are free from non-halal component. And due to the advanced technology and science and technology in food and pharmaceutical industry has led to the use of non-halal component in the food product, for example, pork, lard, and also percent gelatin. We tell about what happened in Indonesia. Indonesia has the Indonesian Act number 33 year 2014 concerning the halal product assurance. In the article, in the fourth article, products that enter, circulate, and traded in the territory of Indonesia must be certified halal. And the halal assurance product is done by Halal Product Assurance Organizing Agency. Is an agency that is formed by the government to organize halal product assurance. And also the certification is done by what we call as LPH, or a product halal examination agency. So LPH is the same opportunity in assisting a government body to examine or to testing the halalness of a product halal. Yeah. So in Indonesia, the certification of halal product is mandatory. So all product, food, cosmetic traded in Indonesia that declared as halal must be halal certified. So basically, what I present today is based on our publication. Yeah. published in Trend in Food and Science and Technology related to review on analytical method for analysis of percent gelatin in pharmaceutical product, in food and pharmaceutical product for halal authentication, and also in International Journal of Molecular Science, the use of a spectroscopy in combination with chemometry for halal authentication analysis, and the last one is based on our paper published in Food Review International entitled Analysis of Pig Derivative for Halal Authentication Studies. So for analysis of gelatin, so we can use some biomarkers. For example, we can use the protein, as we know that gelatin is protein. So we can use protein as biomarker to be analyzed using, for example, chromatography or ELISA, or also based on DNA method using polymerase chain reaction as previous presenter state, and also we can use a fingerprinting profile, for example, using NMR, or also using FTIR spectroscopy. So we divided the method for analysis of gelatin into two categories. The first one is screening, and the second one is confirmatory. For screening, we can use FTIR spectroscopy in combination with chemometry, and for confirmatory, we can use real-time PCR. This is the DNA-based method for analysis percent DNA, or using Lcms for identification of the peptide marker present in the percent gelatin. So as we know that IR spectroscopy is a fingerprint technique. It's rapid and also sensitive, non-destructive, meaning that the analyzed sample can be further analyzed using the different analytical technique. Easy in sample presentation, can be used for qualitative and also quantitative. As we know that the halal authentication analysis is qualitative analysis, because the presence of a non-halal component in any amount is not allowed. So the analysis, qualitative analysis, is the important task. So this is the step for analysis of percent gelatin using IR spectroscopy. The first one is scanning of IR spectra of gelatin from different sources, and then preparation of laboratory sample containing gelatin in food and pharmaceutical formulation, and then the model optimization, analysis of unknown samples. In some, if we are doubtful, so we need the confirmation analysis, we can use the other method, real-time PCR or Lcms. This is our colleague in University of Putra, Malaysia, has analyzed the gelatin sources, but in raw material, not in food or in pharmaceutical product. Our colleague have differentiated between porcine and bovine gelatin from the different sources, porcine and bovine, and from different manufacturer. And basically, this is the FTIR spectra of porcine and bovine gelatin. So in the fingerprint region, especially when 1,500 to 500 centimeter -1, so we can observe the differentiation between porcine and bovine gelatin. However, the differentiation is unclear using the naked eye. So we must use the sophisticated software, sophisticated data analysis that we call as chemometry. So we can use, for example, the unsupervised pattern recognition of discriminant analysis. So if we treat the FTIR spectra of porcine and bovine gelatin using chemometric of different discriminant analysis, so we can discriminate between this is the bovine and this is the porcine. And this is the porcine, and this is the porcine region, and this is the bovine region. So if we have a non sample and then we scan using FTIR spectra and then we treat using discriminant analysis. If the unknown sample fall into the bovine region, so we can deduce that the unknown sample is bovine, and so on. If we have a unknown sample and then fall into a porcine region, so we can deduce that the analyzed sample is coming from porcine. So some scientists also develop FTR spectroscopy for analysis of porcine and bovine gelatin, and also other type of gelatin in a capsule cell. So this is in the product, yeah, not in raw material, but in food and also in pharmaceutical product. For food product, as the model, and the scientists using candies, while for pharmaceutical product, the scientists using the capsules, yeah. And again, because we are very difficult to differentiate between porcine gelatin and bovine gelatin in the food or pharmaceutical product, just only using FTR spectra, again, the chemometric analysis must be done. Yeah. For example, the authors use the principal component analysis. Yeah. And also, this is the cluster dendrogram for differentiation of porcine and bovine gelatin in the food product. So the authors can classify between porcine gelatin in different concentration and also in bovine gelatin. And also, the authors can differentiate between bovine gelatin, fish gelatin, and also porcine gelatin using this is the principal component analysis. Also, this is basically the same in the in all the food product. So the authors differentiate a porcine from different manufacturer, and also in the food product. So, and then we also can determine or identify the porcine and bovine gelatin using SPLC. As we know that gelatin is protein, so if protein is hydrolyzed using acid, so protein is converted completely into amino acid, and then we can analyze amino acid composition in the porcine and bovine gelatin. So, as we know that porcine and bovine gelatin can be differentiated in terms of amino acid composition. This is the basis for analytical principle for differentiation of gelatin using SPLC. We try to differentiate between porcine and bovine gelatin in commercial capsules using based on amino acid composition. And this is the chromatic HPLC chromatogram using I think using OPA, orthophthalealdehyde derivative, to determine the level of amino acid, and we found the composition of amino acid. So it is rather difficult to differentiate porcine and bovine gelatin in capsule cell using amino acid composition. So we use the chemometric or principal component analysis to know which regions correlating with capsule cell containing porcine and or bovine gelatin. And then the method, I think this is the candidate for standard method, is LCMS. By LCMS, we can find the peptide marker which can be used for differentiate between porcine and bovine gelatin. For example, in this paper, mass spectrometric detection of marker peptide in tryptic digits of gelatin. So this is in our paper. We have written the procedure how to prepare the sample before peptide analysis. So sample food or pharmaceutical, and then we digest using a trypsin enzyme. So we can imagine that this is the protein after we hydrolyze, we digest using trypsin. So the protein can be hydrolyzed into peptide with certain molecular weight within the residue, because we use the trypsin. So the residue, amino acid residue, is lysine or arginine. Lysine or arginine. So the protein is cleavage into some peptide with the residue in the terminal. In the terminal, C terminal is lysine or arginine. So we will get some peptide, and some peptide can be used as a marker for identification of porcine and bovine gelatin. So this is the easy principle to be understood. So this is the mass spectra of porcine and bovine. For example, this is the spectra of bovine gelatin, and then we can differentiate between porcine and bovine gelatin using this peptide. So this is the peptide marker for bovine, and this is the peptide marker for porcine. So if we have the product, food or pharmaceutical product, and then we analyze using the developed method, and we find this peptide marker, so we can conclude that the product coming from bovine. And if we have the product and then we analyze, and we can get the peptide marker at least in this table, so we can conclude that the analyzed product contain porcine gelatin. So a number of papers have published the peptide marker for differentiation of gelatin source. For example, this is fish, monkey, and then bovine. Yeah, and to more clear differentiation, some authors use a chemometric. For example, this is use PCA for differentiation between porcine, bovine, and others. So if we have unknown samples, for example, unknown sample, so unknown sample can be classified according to the region of corresponding gelatin sources. And the last method to be shared here in PCR, yeah, as a presenter before has clearly stated that the principle that polymerase generation is a method of choice for analysis of gelatin based on DNA analysis. Yeah, so this is the step of primer design, and then DNA extraction, and then PCR analysis, and then data analysis. So if we, as we know that PCR consists of three main steps: denaturation, annealing, and also amplification. So basically, PCR is DNA replication in vitro. So we have published how to analyze porcine gelatin DNA in a commercial capsule cell using real-time PCR for halal authentication. And this method has been accredited according to ISO IC 17025-2017. So we have the accredited method according to ISO 17025 for analysis of porcine gelatin, because in Indonesia, to be a halal examination body, the laboratory use must be accredited ISO 17025. So the step is optimization of the analytic temperature, and then we specificity test to confirm that the primer is only specific for DNA target, and then we determine the limit of detection, and finally we analyze the commercial samples. From the sample analyzed, this is no amplification peak. Therefore, we confirm that the capsule cells do not contain % gelatin. However, if the negative result indicate that there is no % DNA in evaluated samples, but the question is, does the DNA extraction fail? Because if the samples had if the sample contain % DNA, but the extraction of DNA fail, so DNA is not detected. And is the percent if the negative result, some possibilities, does the DNA extraction fail? Is the % DNA absence? And also what type of DNA present in the sample? Therefore, we confirm that negative samples from % using the primer which is specific for bovine, because the most possibility if the capsule cells negative from porcine, so maybe it is coming from bovine. So the same samples are run using real-time PCR using the primer which is specific for bovine. And this is also published in International Food Research Journal. And finally, the 3 samples analyzed, which is negative using primer specific to porcine DNA, is positive using primer which is specific for bovine. So we conclude that the analyzed samples, commercially available, is coming from bovine. I think this is my presentation, my sharing in this afternoon in Indonesia.
Thank you very much, Professor Dr. Abdurrahman. And now floor is open for the question if anyone else would like to ask anything.
Assalamu alaikum. Thank you very much, Professor Dr. Abdul Rahman. Very nice talk. As a student, I got very much information from your presentation. My question is there any online library for the FTIR spectra of the porcine gelatin for the screening of unknown samples in our laboratories?
Please, not clear because I think the signal is not good. Please repeat or write in the chat room.
My question is any online library available for the FTIR spectra of
gelatin? Okay. I think for halal is in not available, but the common food or common pharmaceutical, I think this is available. For example, gelatin, and also, but for online database that the gelatin coming from porcine, the FTR spectra like that, and bovine like that is I am not sure. But for food or pharmaceutical, the database for FTR spectra are available. But I am not sure is also gelatin included in food or pharmaceutical. Okay, thank you for the question.
Assalamu alaikum, brother. This is Dr. Musharraf. Indeed, a wonderful work that you have presented. I just want to have your opinion because you have a vast experience of analyzing gelatin by various techniques, including mass spectrometry. So, what do you think that which technique is somehow giving a more reproducible result, particularly MALDI-TOF MS? Thank you.
Okay. I don't have any experience for using Lcms. So far, we just only use the real-time PCR and also FAR spectroscopy. So I am so sorry for this. But in conceptual framework that the multi-TOF and also other type of MS can be used for identification of porcine gelatin by investigating the peptide. But we have no data regarding for example, the reproducibility to find the reproducible peptide marker, doctor, like that.
Any other question? Thank you very much, Professor Abdul Rahman Saab. It was really very useful because this is an extremely important area. Now, our young next speaker is Miss Marisa Monpe from Thailand. She is uh, very promising young scientist doing her PhD in food and nutrition from Thammasat University. And she has already worked, published a very good publications, and she has good, excellent experience in the halal food forensic laboratories and these analyses. So therefore, now I am going to invite Ms. Marisa Marn P. to please share with us her presentation. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for inviting the Halal Science Center to Chulalongkorn University to share the information and the experience for the laboratory analysis for halal authentication. My topic today is, I'm sharing the screen. Okay. Have you seen, do you see my slide clearly? Okay. My topic today is Lcms method for detection of gelatin, porcine gelatin in food and pharmaceutical products. This presentation consists of the introduction, which is the background information of the gelatin and the LCMS technique, and the method of the detection of porcine and also bovine gelatin using LCQ trap MSMS, which is the in-house method that we developed. Finally, is the application of the method in the food products for halal authentication. Now let's start with the background information. Gelatin is a protein-based hydrocolloid obtained by partially hydrolysis of collagen derived from skin, bones, and connective tissues of the animal by treating with alkali or acid. Gelatin has been used widely in many industries as a, especially in food as a food ingredient. Gelatin is a solid that is a translucent, colorless, and flavorless solid. The functional property of the gelatin, I think we already well know about the gelling agent property and also the stabilizing agent and thickening agent. It also have the functional property such as a texturization, emulsification, adhesiveness, sedimentation, and also encapsulation abilities. So gelatin has been used widely in many industries, especially in the food industry, such as in the dessert, jelly, gummy products, and also some of dairy products such as yogurt. In addition, it's also used in the pharmaceutical products, mainly in the capsule, either hard or soft capsule manufacturing. In addition, gelatin also has been used for for the non-food applications such as cosmetic engineering and also the forensic. The source of gelatin raw material can become from the cattle, pig, which are the main raw material source, and another alternative source are fish and also chicken. Gelatin can be classified into two types according to the conditioning process or the process of hydrolysis. The first one is type A gelatin, which produced from the acid treatment. It's commonly extracted from the porcine skins and bones of the beef hinds. The second one is the type 2, the gelatin that produced from alkali treatment, commonly extract mainly from beef-based raw material, but sometimes we can find from the pork raw material as well. This figure shows the hydrolysis of collagen to gelatin. As we know that collagen is the protein in the cognitive tissue of the animal, the structure is like a triple helix of polypeptide chains. And after it was passed through the alkali, either alkali or acidic process, it will become to gelatin, which is a single strain poly, polypeptide. Uh, it was found that the gelatin amino acid sequence varied depending on the source of gelatin. So let's take a look at the composition of each type of gelatin. Gelatin consists of the amino acids. Uh, most the most abundant amino acid in gelatin are hydroxyproline, proline, and also glycine. Especially the hydroxyproline. Hydroxyproline is a specific amino acid for gelatin. So this amino acid has been used for detection and quantification of gelatin in the sample. When we see the different type of gelatin, for example, you can see that uh, asparagine and glutamine cannot be found in the type B gelatin because they already changed to aspartic and glutamic acids. Why it still be found in the gelatin type A? Now I'm going to talk about the source of gelatin. This figure shows the global gelatin market by raw material in 2016 and the expected growing in the future. You can see that the green one of pig skin is the most raw material source in the largest section in the market share, and is expected to grow in the future in the forecast period. While the second large segment is the bovine hides, and another one is the gelatin made from bones. This is interesting because it's growing fasting in the future. The high quality gelatin mostly come from porcine and bovine gelatin source. That's why we can see the large segments in the global market. And the source of gelatin is an important issue. It is true because, as we know, Muslim and also Jew are prohibited to consume any porcine originated food, while Hindus are prohibited to consume cows. For the Halal issue, the source of gelatin is a constant issue in the Halal authentication. So many, however, most of food labels do not indicate the species origin of gelatin. So that's why many researchers try to develop the method using many techniques to differentiate or identify the gelatin source. As Dr. Abdurrahman already reviewed about that, this slide shows some of techniques that has been researched or used for differentiation gelatin source, such as Fourier transform infrared spectroscopy or FTIR, liquid chromatography, mass spectrometry or LCMS, or LC coupled with tandem mass spectrometry called LCMS, enzyme link immunosorbent assay or ELISA, and also polymerase chain reaction, which is the DNA-based method. And focusing the technique of LCMS, Liquid chromatography tandem mass spectrometry or LCMS is an analytical chemistry technique that combines the separation of capability of liquid chromatography with the high sensitivity and selective mass analysis capability of the triple quadrupole mass spectrometry. This figure show the some example of the LCMS, such as the ultra high performance liquid chromatograph coupled with Q-trap or Q-trap mass spectrometer. What is the mass spectrometry? Mass spectrometry is an analytical technique that determines the molecular weight of the chemical compounds by separating the molecular ion according to their specific mass to charge ratio or MOC. This figure shows the component of mass spectrometer with the first part is uh, it needs a sample introduction like GC or HPLC, and after introduced to the MS, the compound that we interest will be the compound will be ionized to be the ions, either the positive or negative charge, and then the MOC of them will be further analyzed using mass analyzer such as Q-TOF, Q-trap, ion trap, etc. And finally detect the select MOC that we interest. For the MSMS or a tandem mass spectrometry is a MS technique that allows for production and identification of the fragment ions from a select ion. The fragmentation process called collision-induced dissociation. You can see this figure. After ionization of the compound, it will be the MOC will be separate and show you as a MS spectra. This is a parent ion of the compound that is specific of each compound. But for MSMS, that parent ion will be further fragment to be the smaller one called daughter ions, and we can get the mass spectra of the daughter ion. So the couple with the MS spectra for MS1 profile and also MS2 spectra profile give us more selectivity to identify the compound that we interest. Um, this slide show that I would like to talk about the analytical method for gelatin analysis of our organization, which is the Halal Science Center, Chulalongkorn University, uh, from the routine quantification lab to development of the method to identify the gelatin source. Basically, we have the uh, spectrophotometric method, which is the hydroxyl method for detection and quantification of gelatin, which we usually use in the laboratory services. Uh, but this method cannot identify the source of gelatin. So our researcher developed the LCMS method to identify the to differentiate the gelatin source. And at that time, she found that she was successful to differentiate between the mammalian gelatin and fish gelatin. However, the among mammalian gelatin, like the pig or the cow, porcine and bovine gelatin cannot be distinguished by using LCMS. Then, a few years ago, our researcher developed the method by using LCMS to differentiate between porcine and bovine gelatin, and we also successful to differentiate and find the markers of the bovine and porcine gelatin. So now let's move to this method, the detection of porcine and bovine gelatin using LCQ trap MSMS, which is an in-house method. Um, because of the data of about the development and the validation is still in place. So I cannot show you the clearly picture and the more information. But I will tell you roughly that this technique has been developed by using LC, ESI, QTrap, and SMS. Uh, and we can differentiate between pulsar and bovine gelatin by using many steps like principal component analysis to differentiate and then finding the specific markers of either bovine and pulsar. And this method is, we can call it quite a rapid method because it's direct digestion, no extraction is needed. And also and this method also allow us to detect both pulsar and bovine gelatin markers and also applicable in food sample. The scope of method is the sample is in the group of gelatin containing desserts such as jelly, gummy, and marshmallow, and also hard capsule and soft capsule. The principle of this method is the polypeptide chains of gelatin will be uh, hydrolyzed by using trypsin enzyme, with will be cleavage at the carboxy uh, C terminal end of the lysine and arginine, and brings up and leading to and change the polypeptide to the shorter chains. Uh, Peptides, and the peptide marker also include at the in the that peptide mixture, and then they will be further analyzed using LCMS analysis. Uh, there will be like separate by using HPLC column and select and fragmented according to the specific M over Z using multiple reaction monitoring mode, which allows to uh, analyze the multiple daughter ions at the same time from the one or more parent ion. After data analysis, uh, uh, the sample that we can detect all five pulsar peptide markers, and also the for daughter ions can be reported as a pulsar gelatin detection in the same way as the bovine gelatin. Uh, this slide shows the briefly process of the method. Firstly, we need the sample preparation and then direct tryptic digestion. We incubate it overnight, and then the sample will be further centrifuged and filtered before injection through the LCMS. After the analysis, LCMS will provide us the MRM chromatogram and extract ion chromatogram, and we need to analyze to select whether it contain marker of marker for pore or not, and each pore marker it it have to contain another four daughter ions to 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 complete that marker, like this figure. This figure in the green cycles show the extra ion chromatogram of one pulsar marker, which illustrate four daughter ions. If we found this and we found another four pulsar marker, so we can say that is this sample contained pulsar gelatin. This method was tested for the specificity in the gelatin, many kinds of gelatin, and also the hydrocolloids that come from polysaccharide, because the hydrocolloid also be used in the dessert or gummy or jelly product, and the product that need the thickener agent as well. So we test is in them. this table shows some data of our all experimental data. uh, it was found that except for porcine, except for porcine and bovine gelatin, uh, no positive result found in other gelatin, like no positive result found in fish gelatin and also polysaccharide hydrocolloid. So this show the specificity of the marker we developed. The right your right hand side show you the accuracy of this method conducting in the bovine gelatin containing products like some candy, gummies, and marshmallows that indicate in the label that this is the made of the bovine gelatin. After after analysis using this method, we try to indicate the species origin in them, and we found that 100% of all products of the bovine gelatin containing product show the result of the detection of the bovine gelatin, not uh, not show the porcine gelatin marker in that product. And I'll already show you about the method, but how can we apply this method for Halal authentication? I will talk about this in this section. For the application of the method for Halal authentication, uh, actually LCMS method quite is the advanced method and quite high cost for high cost for analysis because the instrument is very high uh, price. And so we need the screening method for the detection of the gelatin first. At the Halal Science Center, uh, the workflow of the analysis is when we have the doubtful samples, we will analyze using the screening method for gelatin detection and quantification first. The method we use is the hydroxyl method. And after we found the sample with the positive result, which means that it contains gelatin. So, and if we would like to know whether the source, if you would like to know the source of gelatin content in the stat sample, we will further analyze using the advanced methods for identification of gelatin source by using LCMS. And these are, I will show you the application of the method in the real sample. This is a part of the of research we can call like a mini review from our scientists uh, that present poster in Thailand Halal Assembly 2019. They conduct the detection of bovine and porcine gelatin peptides in food product and capsule. The product consists of the hard and soft dairy supplement capsules and also jelly and marshmallow. All products already all products were analyzed firstly by using hydroxyproline, and the sample that found significantly amount of gelatin will were further analyzed using SMS technique that we developed. The results show that for like for example the jelly, uh, most of jelly product are porcine gelatin, and the total and for the total of the total of product, bovine gelatin was the most abundant in that sample, uh, followed by the mixture of bovine and porcine gelatin, and also uh, porcine gelatin alone. Some of them also cannot be detect either bovine or porcine gelatin, which may be because of they use another source of gelatin in that product. Okay. Let's to wrap up all of my presentation, I will show you a video clip that presents the flow or the workflow of the analysis of gelatin content and the advanced detection of pulsar gelatin in food and product for Halal authentication from the Halal Science Center, Chulalongkorn University. I will share you by using another program. ต้องสต็อปนี้ก่อนเดี๋ยวมีต่อเดี๋ยวนะเราเปิด oh I need to stop this first and can you shareเขาเห็นกันใช่ไหม The first method is a hydroxypyrrolene method, which is a spectrophotometric method to quantification of gelatin in food sample. It's a quantitative method. Firstly, the sample will be hydrolyzed using sulfuric acid and precipitate. After hydrolysis, the hydrolyzed hydroxypyrrolene will be further oxidized using the oxidant to produce the reaction, and the coloring agent will be added. The change of color from the reaction will be further detected by using spectrophotometer. However, this method cannot indicate the species origin of the gelatin. So we developed the Lcms method to detect the species origin of gelatin. Firstly, the sample that we detect the gelatin will be further analyzed using LCMS with the Q-trap mass spectrometer. This is an advanced method that analyzes the compound according to the mass to charge ratio. Firstly, we will direct digest the sample by using trypsin enzyme overnight. The trypsin will be cleaved the peptide chains at the position of lysine and arginine to produce peptides. This is the instrument that we use. After incubation with trypsin for 16 hours, the sample will be first centrifuged and filtered before injection to LCMS. We will get the MS chromatogram from this analysis. And we need to find whether it contains porcine marker or not. We need to find each marker and find more that whether that each marker contains the four daughter ion or not. If we found all 5 markers with their 4 daughter ions, we can report the detection of porcine gelatin contamination in the sample. Okay, we'll go back to our the slide presentation. Stop share and no, so I have to share it again. Oh, so this all of my presentation today. Finally, I would like to thank to Associate Professor Dr. Vinay Dahlan, the Director of the Halal Science Center, Chulalongkorn University, and Assistant Professor Dr. Sukit Sirikwan-Pong, the Director's Consultant for Laboratory, and all scientists of HSCCU. Finally, I would like to invite you all to join or to participate in our conference, which is the International Halal Science and Technology Conference to 2021 or ISATEC, which is the virtual conference which will be held on this December. And for those who have any scientific research, uh, we would like to invite you to submit your paper uh, in our in our conference, and the selected paper will be further published in the scientific international journal. For more information, you can visit. Please visit at this website, www.exatech.com. Thank you.
Thank you very much, Ms. Marisa. This was a very, very useful sharing of your knowledge and expertise and research. And now I can open this for the question from the participant, please.
Thank you so much for your detailed presentation. I just want to know, in one of your abstract, you showed the percentage-wise breakdown of the different markers. Could you please let us know that what was the technique or the mechanism for this percentage-wise breakdown?
Thank you for your question. Could I repeat the question is that you need me to explain more about how we get the markers?
So the markers, I'm a bit familiar with the LCMS to detect the marker, but I just want to know how we can calculate the percentages that you have showed in one of your abstract in the bar chart. So yeah. I mean, if someone want to detect it quantitatively, so what's the
You mean
technique?
this?
Yeah, yeah.
Oh, this is the survey research from our scientists. I would explain that that each bar chart, uh, let's say for the hard dietary supplement capsule, the percentage one is like the number of all sample will be transferred to 100%. Each bar chart represents 1% of the number of the product, and like for the first bar, the 54% means that the 54% of all products numbers con contain bovine gelatin.
Okay, thank you.
Is it give you the clear information? Does this answer your question? So this means the percentage show is the percentage of the number of products. Mhm.
Assalamu alaikum. Thank you very much for your nice talk. You have used spectroscopic techniques for the quantification of gelatin, and then you used tandem mass spectrometry for the identification of species of origin of that gelatin. My question is that are there any clue or information in the spectroscopic technique on the basis of which you have decided to go further for the mass spectrometric technique, and are no any other information?
Your question is that what is the clue of analysis by
As
using LCMS?
spectroscopic technique on the basis of which you have decided to go further for the mass spectroscopic technique.
Or why, like after screening method, how can we consider to measure the LCMS, like a further analysis, right? This
Yes.
is your question. Uh-huh. Uh, Actually, with it's for the sample, actually it's depend on the consumer, that whether they would like to know the source of gelatin or not, or they would like to know only the amount of gelatin contained in that sample. But for in the case that we would like uh, to use the laboratory result for Halal authentication, uh, yeah, sure, it should be analyzed the gelatin source. Um, After screening method, only sample that we found the positive result. That means the sample only the sample that contain gelatin will be further analyzed for in the advanced method. But because if you analyze all of them, including the sample that not contain gelatin, so it's like it's waste of your chemicals and waste of your time, right? So it should be screening first and select only the positive one. Yeah. This is
Thank you.
clearly. Thank
Yes.
you.
Thank you very much, and Ms. Marisa and all participant and contribute, and Professor Musharraf Sahab and Comstech and was. Acha, oh, there is a one
Assalamu
more
alaikum.
question, please.
Assalamu alaikum, Madam Marisa. There is two questions. First question, you have mentioned hydroxyproline is an important marker in the proline and bovine gelatin uh, differentiation. First of all, I have question, do you have studied the isotopic pattern of the hydroxyproline in the tandem mass spectrometry, which can differentiate between the proline and the bovine gelatin?
Yeah, thank you. The questions. The questions is that can we differentiate the hydroxyproline of
Hydroxyproline isotopic differentiation pattern in the tandem mass spectrometry.
uh, actually, hydroxyproline is the smallest unit of the polypeptide chain. This is the amino acid, right? So it's too small to. Actually, hydroxyproline either in bovine or porcine or fish is also the same one, which is the hydroxyproline amino acid. But different things of the marker from bovine or from either bovine or porcine are the like the composition of the peptide marker chains that consisting of uh, a few amino acids. This make like how to say uh, the different source of gelatin after we cleavage them with trypsin enzyme, they may uh, yeah, the cleavage, the hydrolysis is the same way. But uh, if you can remember that I showed the type of gelatin, each type of gelatin consists of different amino acid sequence. or amino acid composition show the result or the products from tryptic digestion will give the different structure or different amino acids composition. And this difference in amino acid composition means that they also have the different molecular weight and different mass to charge ratio. And
Uh, Thank you. My second question is about the processed food. Can your method for the LCMS can it is applied for the processed food for the differentiation of the gelatin?
yeah, for the processed food, we have to classify like the scope of that processed food. For this method can rely on the product type that in the group of dessert or jelly gummy that not contain all the source of not contain the very complex of the ingredients. Like many ingredients of this kind of products are gelatin or other kind of gelling agent and sugar and color and some of another thing. But if for the sample that contain very complex of the ingredient, we need more validation, and it need more sample preparation first. It need to be extract for the gelatin first, and then can be further analyzed using the same step. And there will it will also be need to validate the detection in that group of samples as well.
Thank you very much. And now we are going to have a short tea break, and we'll be back at 11:10. And please, there is an attendance sheet outside. Please put your attendance, and thank you very much.
Thank you.
ये इसने कॉन्सेप्ट के नाम से कहा
ये कौन सा
ये बस